I mean, there's a really weird mixture here in the sense that normally when I give a talk to a general audience, I avoid all the technical aspects and all that. But this audience knows the technical aspect more than I do. So I have to switch roles somehow and talk about the things that are non-technical that you don't know. So that's most of the thrust, so you get a sense for what it is that we do in there with all these ferrets and the complaints about the smell all the time. So there's a ferret. This I found in a cave in the Pyrenees on the border between France and Spain, and they were doing experiments. 13,000 years old drawing. It's unbelievable. Looks like a ferret anyway. But it was the size of a ferret. It looks like a ferret, so it's a ferret. But what I'll talk about is not really the ferrets, but this kind of a scene. That's what we call the acoustic world. And I don't think anybody here doubts for a second that you can be plopped somewhere in there, and assuming you survive the cars and all that stuff, that you can perfectly communicate with anybody there and talk, and all this incredible noise, you can just focus on one source and ignore everything else. How on earth is this doable? And we have really no algorithms that you can just buy and actually have anything approximating the incredible abilities of humans and animals. Frogs, when they mate, have you seen a scene when you have hundreds of frogs all yelling at the same time and all that, and a male frog, a female frog can decide exactly which male she likes and aim in that direction, ignoring all these other males that are yelling and screaming. And the same for penguins. So the reason penguins are really interesting is because it's all flat there. It's all white. There's nothing that can tell a mother penguin coming out of the sea where the chick is. And people have studied this, and it's the call of the chick among tens of thousands of penguins, and she aims straight for the... So we're trying to study basically this kind of performance, this kind of behavior, these kinds of... What I'll try to tell you by the end is, algorithms that can do this kind of thing we think are biologically plausible. That's the claim. Okay. First of all, the device that does all this is incredibly complicated, and just to emphasize that this is a project that is €1 billion worth of money centered in Switzerland, but it's all over Europe. And it's called the Blue Brain Project. And basically, they're trying to basically model. This is a cross-section of the brain. And one of those things here look like a column. And they're trying to capture as much as they can about one column in the brain. There are billions of these columns, millions. And each column has literally many, many, many millions of cells connecting. And so the kind of detail is really staggering. And that's the kind of complexity you have to deal with. How do you actually even begin to study the kind of complex task that I just described in a complex structure like this? And one thing you should know before I start, and I'm going to show you this poster that we did for the ISR celebration, the 25th or 30th, is that when you study this kind of a problem, it doesn't come in a given department. People who worked on it in the lab come from just about any background you can grab. Some people are experts in physiology and biology and all that. Some people are machine learning. Some people do psychology and experiments like psychophysics, where you put people in play sounds and try to figure out what they detect and what they don't detect and all that. And others are hardware people. Every single one of them contributes. Some people train animals. Every one of them contributes a view which complements all the other views. Without it, you really are just going half blind. And you can make terrible mistakes. So you have to be able to communicate across. And so all the students and the postdocs and faculty that work in the lab really try to communicate across all of these disciplines. So what I will describe, in order to go back, remind you, I'm always going to go back to this problem of how do we navigate a complex auditory scene? How do we segregate and look at sources? I'll talk first about, well, how is sound actually represented in the brain? There'll be 3 slides on that. One of the key ideas is that helps us do this is that when you actually are in a complicated scene and all that stuff, you're constantly making decisions, you're constantly paying attention, you're tracking sounds, you're doing all this active engagement. Without it, you can't do anything. And when you do this kind of active engagement, you're constantly adapting your brain, changing it literally with networks, that reconfigure and all that in order to do the task. So this is idea of rapid adaptive properties of the brain that allow you to basically adapt your machine to the environment you're at. And without that, again, you can't do much. And finally, I'll talk about temporal coherence principle, which is principle of how sound needs to be in order for you to segregate it. So this is not eye control. This is really what the environment gives me, and I should learn how to make use of. So, first of all, the auditory system, like other sensory systems, is you basically have this sort of sensor and then a whole lot of different areas that the sound is converted to electrical activity, and it travels up the pathway all the way up to the cortex. The cortex is the part of the brain that you see when you open up the skull and you see all these sort of nice jagged things and all that. And by the time the sound arrives there, it has gone through many, many stages of transformation. In the auditory system, like about 5, at least 5 of them. 5 synapses, 5 connections before you get to the cortex. And we're recording in the lab here in the auditory cortex, and the input is sort of what comes out of the cochlea of the ear, which takes the sound and breaks it up into like a piano keyboard. And then all this information is conveyed all the way up to the cortex in a fairly organized way. And what we think is going on here is that all these different structures extract different aspects of the sound and then in parallel feed them up to the cortex. Yeah?
This 5 level, is it ferret or humans?
No, this is it's actually pretty similar, whether it's ferret or humans. Pretty much mammalian cortices all have this kind of plan. And you can go to birds and to frogs and all that. They might drop 1 or 2, and so, but it's pretty much very similar overall organization. That differs in details. Like, for example, here, these 2 nuclei, these areas is the first place where sound coming from your right ear and from the left ear are compared. And there are 3 different regions, or 2 different regions, large regions here, that do this comparison. But depending on what animal and the nature of the cues can vary, the size of these structures can differ enormously. So in a bat, for example, which is a very high frequency animal, it tends to have a huge representation of this LSO region compared to the MSO, whereas in a low frequency animal, or an animal that uses the timing cues between the two ears, it has a much more enlarged region of the MSO and much more in the LSO. So that kind of difference reflects the nature of the cues available to the animal. So some regions we think are involved in extracting pitch. What other things do you know about sound? Timbre, that tells you this is a violin and this is a piano and this is, or different voices. With, this is the timbre. Whatever it is the quality of the sound. Loudness, location in space. A lot of features of sound are extracted and shipped off to the cortex. Okay? This region is, to first order, fairly conserved, meaning you're kind of born with it, you adapt it a little bit during development and all of that, and then it's possible that they do show some of the things that we see in the cortex, which I will talk about, rapid adaptive, but really they're much less adaptive than what you see as you go up further up the chain in the cortex. This is how we do the experiments to study in the animal. So these ferrets, they're generalist animals. They like to hear sounds. They go hunt in dark tubes, and they're ferocious hunters, hunting rabbits and all that. And they're very clever animals. You can train them on many different behaviors. They're the ones that laid out the electrical cables in all of European cities. In the 19th century, they didn't have robots that go around in the queue. So they put a ferret, they tie around it, and they said, go and come out the other side. And the ferrets run down the tubes, and they run the cables. And and they use them in barns, and they bred them actually to kill rabbits, because the rabbits and the rats and the rodents come and eat the grain and all that. And these guys they can chase down a rabbit 10 times the size, and they just not let go until they get it. And American ferrets are endangered species, very endangered. These are domestic ferrets that we get from upstate New York. They're bred for research. And in Europe, we still get them from New York, because nobody in Europe would give you a ferret. But they do have their own native. So these animals are held in a tube, fixed head, so that way they don't move. And then you can put wires in the brain, or image, or do whatever you want to do, and record the electrical activity in the brain as you play sounds. And these animals are trained in advance to respond to sounds in certain ways. I'll show you examples. And they can report to us whether they can hear the sound or not. So we know they detected or they discriminated and all of that by licking the spout of water here. They're thirsty. They're not doing this for research. You have to make them a little bit thirsty so they get all their water during the experiments. And then you record the electrical activity. So you get about a couple of hours worth of work from a ferret before they get enough water and they say, that's it, I'm done. It's a good life. They live with us for many years. We have some ancient animals, like 7 years, 6 years. They cost a fortune. Just we're incompetent about using them. When you go up to the brain and you open up the skull, that's what it looks like. And this is the area of the brain that is considered the auditory area, where sound activates the electrical activity in the brain there pretty vigorously. And if you blow it up, you see that, in fact, it doesn't come marked like this with labels. But we know from anatomical studies and all that these regions are the regions that receive the input first directly from down below. And then these guys are connected to a secondary area, heavily interconnected. And then a secondary area is connected to another area, tertiary area, and so on. So all these regions really have a purpose. We don't know what, but that's what they're trying to study.
Front, right?
The what?
The nose?
The nose. So the nose is this way. Yeah. And this region here is called the frontal cortex, or the prefrontal cortex, responds also to sound, and in fact, the whole brain responds to sound. It's just that how strongly is the response. But we are studying this region and this region. And in this region, the regions respond when the animal is engaged in really much what we call executive functions. They make decisions, they pay attention, they, that kind of thing. They don't just respond haphazardly. Like in this region, when you're asleep, you get good responses. And when you anesthetize the animal, you still get good responses. But the responses start getting weaker and weaker, and only modulated or strong when the animal is engaged in behavior. So that's why you really need to study these animals, not just in an anesthetized or asleep, or even awake but not doing anything. You need to engage them in behavior, like kinds of cluttered scenes I'm showing you. Would make you don't see much. I'll show you. You only see it really when the animal is engaged in behavior. So you have to train the thing. And that's why we have to do experiments in humans, because to train a ferret to separate two voices, two speech people, two people talking simultaneously, it would take my lifetime. But to do it in a human, all of you do is stick them in a room and say, listen to the male or listen to the female and tell me what they're saying. But how do you put an electrode in the human brain? That's the problem. So if you have volunteers, we can pay really well, at least $10 an hour. Don't laugh, it's a lot of money. Okay, so what is the cortex for? I'm going to show you first really basic pictures, obviously, because just to give you a feel for what you find when you go and stick electrodes in the brain. I'm going to tell it to you from a mathematical point of view, because most of you would probably understand that much better than the biology of this. But this is the spectrogram that comes out of the cochlea of the ear, just the very first neural activity coming out. And that, by the time you get to the cortex, the major transformation, aside from extracting pitch and timbre and location and all that, is what we call the spectrotemporal analysis. Literally what it is that if you look at this as an image, one thing you could do is do a two-dimensional, I'm going to use the wrong terminology first and then I'll improve it, do a two-dimensional Fourier transform on it. And that gives you an idea about how fast things vary along this dimension. We call this is the time dimension. So how things vary means modulations, temporal modulations. And along this axis, you see there's a peak, valley, peak, valley, peak, valley, and so on. These are spectral modulations. So if you do a two-dimensional analysis on that, it tells you the spectral and temporal modulations. It turns out this is a very important aspect to describe what a spectrogram is doing. So in short, what I'm going to tell you is that mathematically the closest thing we can come up with for what the cortex displays is it does a two-dimensional wavelet transform on a spectrogram, which is already a wavelet transform of the acoustic stimulus. But a two-dimensional wavelet transform means that you take the time axis and you discover you do a rate analysis on it. Okay, you create channels that are fast and channels that are medium and channels that are slow. And you go on this axis and you do the same thing but on the spectral axis. So channels that are broadly tuned on the frequency axis, channels that are medium, channels that are narrow. And all of these two dimensions of bandwidth and rate are jointly done. And so you get a cube that is evolving over time. So this cube is like a three-dimensional movie telling me at any given moment in time how fast are the different modulations. What are the modulation content of the sound? Both along the temporal and the spectral dimension, and along different frequencies, different frequency channels, and how that evolves as a function of time. This is pretty much what the visual system would do. Actually, I'll show you a picture. If you look at the spectrogram and you analyze it with different kinds of spectrotemporal channels, it's no more and no less than saying these kinds of cells in the brain would look at the spectrogram and see this picture. They kind of look over broad regions in the spectrum and over slow time, and so they lose all the resolution, but they give you a very different view of the spectrogram. This one gives you this view of the spectrogram. These cells give you this view of the spectrogram. It's like if you're looking at a painting and you want to look at a painting and understand it. One really good way of doing that is to come up like far and look at all the broad, smoothing. You get broad view of what the different things and the painting are conveying. And then you can come up real close, the other extreme, and you see the brush strokes. And you get detail that is not available to you when you look far. So that's called multi-scale view. And you can do it both in time, and you can do it both on space, in a vision. Okay? The same exactly as the the auditory cortex does. It turns out that voices, instruments, just about any sound has unique content that tells you the nature of the sound with the spectral and temporal modulation content. That's why a piano, so if you look at like the content of a clarinet, that's the pattern of a clarinet, that's the pattern of a violin, that's the pattern of a piano, that's an oboe. These would be different activation patterns in your brain, and those tell you whether this is the timbre of a piano, that's the timbre of a violin, and so on. Okay? And speech, you can imagine. So if you take, these are actual recordings from a ferret brain, from about 100 cells in a ferret brain, and you look at the activation patterns in the ferret brain for P, ta, ka, ba, da, ga, da, 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 and you see a Persian carpet. But that's how complicated it looks. But all the information is there. And you can train ferrets actually to discriminate between different, I'm sorry, this is actually not showing the data for discriminating behaviorally, but this is how humans, for example, discriminate between pa, ta, ka, fa, sh, and that's the confusion matrix. And you see the neural confusion matrix from a ferret looks actually quite similar in structure and the way. So humans confuse pa, ta, ka, and noise in a very similar way to, they never confuse pa, ta, ka with fs, sh, and neither does the neural data from a ferret. And we can actually record in a ferret brain. So this is work that that a student here, Nima Mazgarani, did many years ago now, 5 years, 10 years ago. And he recorded in the ferret brain for speech. Before he did that, he went and recorded from 100 neurons. He recorded the forward path, the transfer function. He took 322 or 631, or 630. Okay? He measured basically the forward path, transfer function of these neurons, literally. And then he played speech. He didn't know what it was, but he took the activation patterns and he played them backwards through what he characterized. So he reconstructed from the ferret brain activity of 100 neurons what the sound is like, what information is in the sound in a ferret brain. And I'll play you that. That's the input.
Bridges, tunnels, and ferries are the most common methods of river.
Oops. And that is what he reconstructed.
You play together.
Bridges, tunnels, and ferries are the most common methods of river.
This is what he played in.
Bridges, tunnels, and ferries are the most common methods of.
And this is what he reconstructed from the ferret brain.
Ridges, tunnels, and ferrets are the most common methods of
Completely understandable. And this is only 92 neurons, not 92,000, 92 million, because we don't have the ability to actually do these kinds of recordings. Okay? So,
You think the identity of the speaker is preserved still? From reconstruction?
well, a dog owner would assure you that the identity of the speaker is preserved. So with a ferret owner, I don't know. It's amazing what dog owners. Okay, so now let me get to the second point, which is that your brain is adapting on the fly all the time. I would say even if you didn't like it, it's changing. So I'm going to illustrate that for you with this amazing stimulus. And then it's called the shepherd tones illusion by colleagues in France who worked on this. And ferrets actually, anyway, let me just play you. So I'm going to play you as the following. A bunch of sounds, doot, 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 like they sound like tones. And then a little gap, which can be, by the way, quite long, many seconds or even minutes. And then two test tones, doot, doot, or doot, doot. These two test tones, they change in pitch. You tell me whether the pitch is going up or down. Okay? First. How many people say it's up? Pretty much everybody. And this? Down. So I'm going to play you just the last bit here again. Oops. Down? Yeah, actually, okay, let me just tell you, in the interest of time, the last two sounds are identical. Huh? If I play you one of those tones just for 20 milliseconds, I can bias you to hear it up or down. So somehow, your brain, by just listening to a context for a really short time, can completely change state and hears the same exact sound either as up or down. It's completely crazy. Yeah.
What's the spectral content of those sounds?
It's what you see. So in this particular example, it's a complex of tones that are octavely spaced, and the second one is exactly halfway between the two.
The coefficients didn't change?
The what?
The coefficients?
Identical. The two stimuli are identical. I can play them, the last two in the one which sounded up, and if I play you the down first and then I play you that one, it sounds down. Whatever I induce in your brain, it stays there for a period of time. If you lose kind of focus and all that, you lose it.
But if we just played the testing part, not the biasing part, would we hear them exactly the same?
Half people hear it up, half people hear it down.
Even though they're still exactly the same tone?
Yeah.
So it just, the context...
The context shifts you up or down. Exactly. That's identical to a lot of folks, for the benefit experiment of Arda and Alga. In fact, it's no different.
What do you need to do to adapt to a particular speech?
Exactly. Except there also, it turns out that with studies that you don't need much of a cue to shift your format and make you hear the identical stimulus as either da or ga. And people swear they hear it as da or ga. Yeah.
Does an individual person have a consistent bias or is it random?
They tend to have a consistent bias. But, the traditional studies like this tended to be, oh, you are from England and you heard a lot of Beatles, and so you tend to hear it down. And if you come from Canada, you hear it up. And if you come from this, it's just BS. It's some random things in your brain, who knows. But humans, they like to look at a sky and make a story out of it. So this same student, Nima, who worked on the ferrets, actually he decided much better to work on humans. He got fed up trying to train ferrets doing stuff. So what they do in these experiments is these are people who have epileptic seizures that are severe enough that they have to be operated on. So they open up their brain, they stick electrodes on the brain. You're seeing the electrodes on the brain there. And they sit in the hospital for a period of time. And they wait till the seizure to come so they can see where it starts and where it ends and all that. Where's the auditory areas of the brain? And then the surgeon would come there with a scalpel and just cut out, literally. That's the surgical operation. They cut the region of the focal region where these seizures begin. And that will cure them. It's very nice. I don't know what else you lose, but in any case. But they want to avoid losing the auditory areas because it's important for language and all that stuff. And they don't like the Beatles anymore afterwards. They're totally... So what he did is while these people are sitting in the hospital, they're bored. He said, "Well, we'll do experiments with them." And so he trained. He went to the hospital with the surgeon, and they get the permission with them. Okay, listen to two people talking, male and female, and then at a certain cue word, switch either to the male or the female according to what the cue word tells you. And I want to see what we get in your brain recordings, electrical activity in your brain. And then he applied exactly the algorithm in the ferret to these recordings. So he reconstructed the spectrogram of the sound that was generating the activity in the brain. And these are the reconstructed sounds from pure female, and this is a pure male voice. Okay? So this is the female alone voice, and there's the male alone voice. And reconstructed from the electrode activation, that's what you get. Now he mixed the two sounds, and he asked them to attend to the male and the female, and then he reconstructed it. And what you see here is the activation patterns when people attend to the male or pay attention to the female. Completely different. Within 100 milliseconds, the activation pattern in the whole brain changes and becomes to reflect either the male or the female. How on earth is this happening? That's the key. But this demonstrates that in fact the same identical stimulus by attending to male or female generates a completely different pattern of responses. One last demo.
So the words they're saying are different, right? The female.
Yeah. You have to say different words. That's the third part of the talk. Yeah, they're simultaneously talking. And the task is to report if, for example, you hear, suppose the cue word is tiger. And if you hear the word tiger, then you pay attention to whoever said tiger. And you report the color and the number. And that's how you know that the patient actually was listening correctly. And then you do this many times.
Can you do this if they're the same gender?
Sure.
So does it...
You make more mistakes. As long as you're saying different text, then you can. If there are different timbres also and all that stuff. And so we can talk about this. Yeah. One last demo just to illustrate how flexible your brain is. Some of you have seen this before. It's called the McGurk effect. And this fellow here is going to say ba or da. Okay? And look at him. And after he says it once or twice, close your eyes and hear again what he's saying. And tell me what he's saying. So just to make it simpler, look at him first with your eyes open all the way.
Ba, 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 ba.
Oh, wait a minute. Why is this? This is supposed to be a movie. This is not going to work. Sorry, one second. Let me just see if... You're supposed to look at his mouth. That's the problem.
Ba, ba.
Okay, it's not going to work. The demo actually is if you look at him and you look at his mouth and you see he's moving his lips in such a way as to indicate that he's saying da, 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 da. But what did you hear just now? Ba, ba, right? Because he wasn't moving his lips. But if he moves his lips as it's supposed to do in the movie, you absolutely hear da. You cannot hear ba. So, yeah. So that indicates or shows, yeah.
So the distinction between ba, da, and ga are usually found by the transition of the forms. So
That's because you took the course with your advisor. Just don't believe that.
no, if you look at it, the forms,
You're just looking at the spectrogram. You didn't look at the lips. You didn't look at.
right. So is it possible that one of those tonal effects that the tone entering bit effect that you showed is contributing to the confusion of these three?
Contributing to?
To the confusion of these three things. Because you need to know whether the harmonic is...
Yeah, it's possible.
Like, is it possible that that effect...
No, this effect, it's the identical acoustic stimulus. Sure, your cochlea has exactly whatever you learned in that class. So that didn't change. It's when the visual input comes in, it gets integrated with the auditory input, and the two have a different, different structure to them now. And you read the whole thing. And the whole thing says it's da, it's not ba. And so you ignore the acoustic part that comes out of the cochlea and you shape it according to what the visual is telling you.
Right.
And you can do the opposite too.
Is there a theory about how that gets trained? Because you recognize the
You're
lips moving connected with the sounds only because you've seen lips moving.
Yes.
You couldn't recognize the sounds the first time.
Absolutely.
You put all that together. Just different words that happen to have those different sounds. And
No, you're constantly training every day, looking at people's lips, and you're making this mapping.
How did I know what they were saying before I knew what they were saying? Does that question make any sense?
But they were agreeing. See, he's talking about when the cues are conflicting with each other. The lips are telling you one thing.
That's
the sound
right.
is telling
That's
you
right.
something else. Then what
Reinforcing it.
Exactly. Yeah, it's resolving a conflict because the visual cue is telling you something different than the auditory. Normally in sensory systems, when you have these conflicts, you feel nauseous. Luckily, we don't hear this kind of sound all the time. And in the lab, when we train the animals to detect, for example, sounds moving fast, sounds moving slow, and all that stuff, you see these changes in the responses of the neurons, in the transfer function properties of the neurons. So this movie was made by a graduate student a long time ago with data collected by Jonathan on this ferret that was lucky that you could record. It's not lucky. It's Jonathan, when he records, he gets 2 hours worth of data from one cell and put it together as a movie. And what you see is how this transfer function of the neuron changes. This is actually how we got into this business, these kinds of data. So I'll just show you, and what you're going to hear is a soundtrack that tells you if the animal is behaving or the animal is passively listening. And this picture is when the animal is passively listening. Listening passively is indicated by noise, and when the animal is engaged in a task where they have to detect the speed of a click train, the latency of the response, the time between 0 and where the peak is, that time constant shrinks significantly, as if the neuron is going fast now and is trying to do the task. So there's the... So this is passive. And now it's behaving. What happened? The movies are not playing, I don't know why. But if you had seen the movie, you would have seen this guy move all the way over to here. And then when the passive animal, again, about 90 minutes into this, it goes all the way back, relaxes. As soon as it's engaged in the task, it moves back up again, and so on. This is very annoying. All right, I'm not going to get to say a heck of a lot more, but I just give you a big picture stories here. When you go to the prefrontal cortex, to the frontal area of the brain, You can do experiments where you stimulate there and see the effect there. What we think is going on is that these areas here, the executive function areas, where supposedly attentional effects, the meaning of the sound is stored, affect the responses here by connectivities between these regions, and that's why we see these changes in the transfer functions of the cells and all that. So when you record here, what you actually see is responses that reflect not so much the acoustics of the sound, but rather the meaning of the sound. So for example, you play a sound that is a warning sound or a warning light, and you see big response, very at the onset of the warning. But when the sound, this sounds very strong energy and all that, is a safe sound, no response. It just ignores it. Okay? So by the time we come down the chain from A1 to the secondary, to the tertiary, to the prefrontal cortex, the sound completely changes its representation from simple two-dimensional wavelet transform to more categorized here. Soft categories are formed between the warning sounds and the safe sounds. I say warning because when we train the animals, we teach them to stay away from the spout when, for example, there's a tone, and when there's a safe sound, they can lick. And so that's how we tell behavior. It's a go, no go. Otherwise, they get a shock or a timeout or something. And here is the tertiary area. You see much stronger responses now to the category of warning sounds and more suppressed responses. So it's like it's a progressive classification system going all the way up to the chain. And that's basically what we spend all our time doing, working our way down this chain and up to the prefrontal cortex. Okay? And you have to do this many different techniques, like techniques here with working with Patrick Canold in biology, doing two-photon imaging, meaning you image the activity in the brain of hundreds of neurons simultaneously, and they fluoresce, and you can actually see when they activate. So you don't have to actually go in and put a wire per cell and see, but you don't get as good a picture of the dynamics of the responses. Another technique is fMRI. People do look at the blood flow and all that. Another technique, which we are hoping to bring over here from France, there's a group there that is developing ultrasound imaging of the brain. It's just an incredible method. You can image the entire 3-centimeter cross-sections of the brain, like a whole mouse brain, in one go, very quickly on a bench top. And it's measuring the blood flow changes in the speed. Give if you look at a capillary, which is tiny, you get resolutions of the order of 100 microns. And it's just magnificent. It's going to give us a roadmap of where to look by doing that imaging. And it's very quick. But these machines don't exist yet. We just basically working on a prototype that we get borrowed from the... All right. One other technique is doing EEG. So this is work, those two experts here, and that expert over there. So EEG, Behtash, for example, and Sahar are working on, you record on the outside of the brain from the human, and then you ask questions and you get them in behaviors, just like that experiments that I showed you with the epileptic seizures, except this is on the skull outside. So this we can really do on you guys without even barely paying. Well, $10 is too much maybe. You have the pleasure of sitting there listening to these nice sounds and all that. And one project we're working on actually is play people music, play people this mixture of sounds and all that. And you can, on the fly, based on the EEG, like when you pay attention to one person or another, we can decode the EEG activity and tell you whether you're listening to this speaker or that speaker. So that's possible now. And the EU actually funded a project in a European project across many places to use this method to control a hearing aid. So that way when you go to a restaurant and it's noisy and all that, you can just pay attention to one person, and you beamform on that one person, and then your hearing aid can hear that person instead of hearing everything. So basically you use your,
How many microphones are you using?
well, you can put like a bunch of microphones. You can put, the main difference, you can put it on an iPhone, do all the processing on an iPhone, and then Bluetooth it up to your hearing aid. So there's a lot of technology issues involved in this, but
Oh, I see, you have.
But the difficult problem is to try to actually decode as much as you can of the cognitive things that you're doing with your brain based on the EEG signal. Whether, for example, your expectation of the sound, you can tell that. You can tell about your decision. You can see there's a signal that comes on the EEG that builds up, and I know what you decided. I don't need to actually wait for you to move your arm or do something. I know you detected or not detected, and that kind of thing. So that is quite possible now. And you can build algorithms to do things with these. You can build algorithms to clean up signals and all that by mimicking what the brain does. Actually, again, this guy Nima, who worked on this problem for detecting speech, it's the state of the art now. What this algorithm, this is the way it happened, is this guy from I didn't know he was from the FBI. He came to the office one day and he said, we would like you to figure out, is there a way to detect speech, that this sound is speech versus music? Or so I said, well, that sounds difficult, but maybe it's a possible way. And people can do this. So you have an existent proof. But the way the algorithms used to do it is really very brute force. They look for harmonics that look like voice and this and that. All we did is we have a model of the whole auditory cortex, this two-dimensional wavelet transform, basically, and looked at the patterns of what speech generates compared to any other pattern, and put it in a support vector machine, as we have, and that's it. And that thing works so well. The FBI took it without telling us. They put them on DSP chips. They use them now all over the place. Why? Because they have days of recordings of audio where they used to have these analysts sit there and listen to this damn thing waiting for something to be said. And to mark it so that they can actually hear it. Whereas now, it's just in 1 second they can go through the entire thing and mark exactly where the speech is.
How about if the person is singing?
No problem. It can tell difference between singing and speech. So it depends how you train the support vector machine. So it's basically classification problem and acoustics. That's all.
But you cannot classify that as either speech or music.
Oh, no, you classify it as singing. For example, so you train it as singing category, yeah. And it automatically worked on also speech from TV, which is really amazing. I didn't know that was possible. But it worked right away on speech coming out of TV. Apparently people on TV talk differently than people in real life. And
how about listening to the commercials and maybe this after?
That's now the creative applications. That's why we're still poor in this play. It's good for Spotify, yeah. Well, there's a project, Gemstone, that is trying to tell the emotional content. Actually, they've done it before 10 years ago in Gemstone also, the same exact project. And also there's a project in 408G, project they're trying to classify the genre of different music. So for Spotify, apparently doesn't have, you have to label it. Yeah. Whereas, but it's trivial for people to tell this is, this category or this category or this category. So, and I don't have time to tell you how we can do the algorithms. I just play you a demo of one. This is a demo I just did because this guy in Paris walked in and he said, I hear you guys do all this kind of stuff and all that. Can you do something for my damn hearing aid? I can't hear. So I literally ran the code on it on a speaker and just give you an example of what it produces. So this is in French. So there's a French guy speaking in the middle of all this mess. And shut up. And this is extracting out that one speaker. So it's really easy. It's easy, but I haven't told you how we do this in these algorithms. So, because that's the next part, I apologize. But I'll just give you flavor, okay? So I'll take literally 4 minutes, 5 minutes, and then we can quit. So I can let you go at 5. This is the basic idea of the algorithm and how these algorithms work. So you need to know that the brain is changing, and you need to know also that there's a beautiful, rich representation of just about everything about the sound in your cortex. But one thing is really important about, like I'm talking to you now. When I talk, I move, open and close my lips and all that. If you look at the time waveform of a speech waveform, it looks very impulsive. Okay? Very impulsive. And when two people talk simultaneously, saying different things, they're both impulsive, and they're actually in each other's gaps. Okay? You're with me on this. So, imagine now, just for one voice, going into your cortex. What are you going to see in the cortex? The sound goes in, it gets broken up into millions of channels. All of them are doing what? The one that comes with measuring pitch is going to have activity or energy only when you open up your mouth and generate some sound that has the pitch, right? Up, like in the words. So all these channels that convey pitch, location, timbre, and all that are modulated in time, all at the same coherent way, synchronous way, because they are driven by the same stimulus. Okay? Now you take two speakers and you add them together, and they have different pitches, different locations, for example, and so on. And you look in the brain, or brain model to be precise, and what actually you see is that the signals that come from this speaker are all co-modulated together, and from that speaker they're all co-modulated together, but they live in different channels and they're completely out of sync with each other. Yeah? So you have 1 million channels all being driven, but 500,000 dominated by speaker A, 500,000 by speaker B, and they're out of sync with each other. So the 500,000 from speaker A are all in sync, and speaker B, they're all in sync, but they're out of sync with B. Okay? That's the key requirement that the environment provides for you. Sources are defined as sources because they generate patterns of energy that fluctuate in time in a synchronous way. Without it, you cannot segregate anything. So let me give you a dramatic example, which is similar to experiments we did to test this idea. Imagine you have a choir, from bass to sopranos. They're all singing, 100 of them, singing in unison. Suppose they're saying exactly the same text. According to this theory, that's a single source. It's a complicated source, but it's a single source. If the soprano sings something different and from the bass, then you have two sources. Or if the different groups sing differently, like an orchestra, play different lines, then there are different sources. How do we know that? Is it just because I said it? No, you can do objective testing. So for example, with the choir, if you ask the soprano to make a mistake, and then you ask the subject to tell you, if, when they hear a mistake, who made it? The people are terrible at it. They will say, oh, the bass made a mistake. It's surprising. We tested this not with the choir, but with simpler stimuli, that on paper looks trivial, like a high frequency and a low frequency separated by octaves away and all that. Tell me which one was the one that had a deviant in it. And they can't do it. Because they all get treated as one source. And so when you detect a deviant, it's like you walking in and you say, something different about you today. Did you shave mustache? Did you put glasses on? You can't tell. Because the face, despite the fact that it has different colors and different edges and different this and different that, is one object. Why is it one object? Why is it one object?
Because you always see it.
Because you always see it moving around. You don't see the nose flying off different place than the eyes and all that. So all the pixels of an image are coherent. That's the only reason that you look at that nice, I'm trying to recognize somebody in these pictures. I don't see Andre here, but anyway. You look at that picture, and why do you think there are 4 people there? It's because you see human beings walking around, and you know that from memory. It's the only reason. If you had seen Martians there, I mean, not E.T., but something more weird, you wouldn't know the difference between them and a computer. You think maybe they're part of the same object, because you don't have it in your memory. So that's the hypothesis, that's the theory, that's been driving a lot of the work in the lab in the last 3-4 years. Cost quite a few casualties to try to really get to this theory, to prove it, to prove that in fact what the brain does, it does principal component analysis, so to speak, segregating the channels that are coherent with each other from the others. And how is that done? That's done because the brain evolves these connectivities right on the fly, because scenes are changing all the time. You can't just have principal component analysis on one matrix. You form many different matrices of correlation matrices, and you segregate these different sources, and you can see the evidence of that, but only when the animal is behaving, not when the animal is passive. So that's sort of drives the idea that you need adaptive properties of the brain. You need also rich representation, because if your pitch is the same as, if I don't extract your pitch, there's no way I can do it. Or I don't extract your location, or your pitch, or your timbre, or some other properties, something that distinguishes you from somebody else, then I can't do it. It's a question of labeling, not a question of... So you have all these channels, and you need to label them correctly, as label A and label B. And you use timing to do them. Okay? So that's the big picture. So I can take questions or...